Hello students, how are you? I hope you are fine. In previous session we have discussed about various application of the microorganisms in various different branches. Okay. Today we are going to see certain examples among these different branches under which the microorganisms are used. So today in this session we are going to discuss about the industrial microbiology and the products of the industries. Okay. So here we are going to discuss about the use of microbes in industries. Okay, so we have discussed about it, like we have discussed about the many different industries under which microorganisms are used. Like we have discussed about the first, that is the food industry. Okay, then beverage industry, then in food industry again we can add the dairy industry. Okay, because dairy products are nothing but a food products itself. Okay, then pharmaceutical industry, okay, then biofertilizers, okay, or you can write biological controls. Okay, biological control. Okay, and many are there, like we have also discussed about the agriculture based industries. Okay, so this will come under the biocontrol agents or the biocontrol or biological control. Okay, then apart from that, for production of enzymes, okay, in enzyme production. Okay, so here we are going to discuss about certain examples among this food industry, beverage, pharmaceutical industry, biological control, as well as the enzyme production. So we'll start with the first, that is the food industry. In food industry, what type of examples that we'll study here? Okay, so as we have already discussed, microorganisms, okay, how they can be useful in the food industry. So microorganism, as they can ferment the complex organic food material into simple substances, okay, with the production of a desirable good. Okay, so that is through the fermentation. So food industry, here generally different category of bacteria as well as certain fungal strains are used to preserve the food material as well as to increase the shelf life of the food material, or the food which has been processed can be kept for a longer period of time. Okay, in the market when we go, we can see different types of food products are there. Okay, different type of food products which are canned, which are packed, right, and the manufacturing date and the expiry date is been given, right. So how that food can be preserved for longer period of time, that can be processed under the food processing industries. Okay, so there are many food processing industries, as we can see different brands are available in the market. Okay, of the this all the brands or food product, they are processed under this food industries. Okay, now apart from that, in the food industry we can take an example here as the dairy industry. Okay, dairy industry. Okay, so here in dairy industry, what we see, the production of the dairy products. Now what are the different dairy products? The dairy products are like curd, then yogurt, then cheese, then butter, buttermilk, then we have the ghee, which is very important again. Okay, and many other products are there, like Shrikhand is there. Okay, etc. So these are the different types of dairy products which are being processed and produced within the dairy industry. Okay, now there is a method for production of all these type of dairy products. Now what is that process called? That process is called as the fermentation. Now fermentation means what? See, fermentation can be defined as the process in which complex organic substances are broken down into simple organic substances, okay, and that can be consumed in different forms. That is the fermentation method. And this fermentation, it is been regulated under a particular device called fermenter, okay, and this is regulated with the help of the microorganisms. So see here we have the different, and these all products are produced from the milk. Milk is the primary source for the production of curd, yogurt, cheese, butter, buttermilk, ghee, Shrikhand, etc. So all these products are produced from a single substrate. Okay. So the one which is going to get converted into simple substance is the substrate over here, and these are the products. Now during this conversion, okay, there is a requirement of specific bacteria as well as specific temperature, okay, or other physiological conditions for production of these products. Okay, now let's take an example how these products can be produced in larger amount. As we know that curd or all these dairy products, they are produced in larger quantity to meet the demand of the peoples. Okay, so what microorganisms can be implemented here for production of this desirable product. We can also produce this product within the home or within the kitchen of our houses, but that product will be in the limited amount. To produce this product in the larger amount, the different dairy industries, what they do, they uses the microorganisms, and generally the microorganism which is used for processing of milk is the lab, that is the lactic acid bacteria. Lactic acid bacteria. Now what is the significant role of this lactic acid bacteria? See, generally when we get the milk, okay, from the milkman, what we do, we keep that milk for boiling. After boiling, what we do, we pasteurize it, right? So pasteurization means what? It is the degradation of the microorganism present within the milk, as raw milk consists of the microorganism, and which may multiply and may produce different type of product, again byproduct. But here what we are doing, we are pasteurizing that milk. After pasteurization, what do we do? See, most of the time milk may consist of, the raw milk may consist of different bacteria. So to produce a particular product, okay, that milk will be not desirable, right? So what we have to do, we have to remove all the bacteria. Okay, after pasteurization we have to cool it. Okay, so we will write down the steps for the and for understanding the processes. So first process under the production of any dairy product is the pasteurization of milk. Pasteurization of milk. Okay, after pasteurization, what do we do? After pasteurization, what do we do is we will allow it to cool. Allow it to cool. Okay. So after allowing it to cool, what we will do? We will add the bacterial inoculum. Okay. Now how to produce that bacterial inoculum? See, for production of a desirable product, suppose we are going to produce a curd. So what we have to do is we are going to take a tablespoon of the curd, okay, and then we are going to add into this cooled milk. Okay. So what we are doing here, addition of starter culture. Now starter culture means what? It will be the culture of bacteria such as the lactic acid bacteria. Okay, if the lactic acid bacteria, how to add this bacteria? We can isolate it in the laboratory. Okay, from the milk we can grow onto a particular artificial nutrient medium. See, whenever we speak about the different bacteria or the different microorganism, these microorganisms can be grown under laboratory conditions. Okay, although they are feeding onto the natural medium, apart from that we can culture them on the artificial nutrient medium. Now here the bacteria of interest for the production of curd, yogurt, cheese, butter, okay, or buttermilk, ghee or Srikhand, any kind of product, even the ice creams are also there. Okay, the bacteria like lactic acid bacteria is required. Now what we'll do, we'll isolate the bacteria from the milk. It will be cultured under laboratory. Okay, after culturing under laboratory, what we'll do, we can add that bacteria directly into this cold milk. Okay, that will ferment the milk and will produce the desirable product. Okay, or else what we can do is we can add that bacteria to small amount of milk which will get converted into curd. Okay, and that curd we are going to add as a starter culture into the cold milk. Okay, and after what we are going to do is we are going to incubate it, or incubation. Now for production of dairy product, the incubation time may be from 24 to 48 hours, or it may depend upon the amount of substrate, that amount of milk. Suppose in a home or in houses, what we do, in the utensil we keep the milk, that pasteurized milk, and we add a starter culture, incubate it for 5 to 6 hours or maybe for 12 hours. After that we get the curd, right? But here in the industries, as we know, the industries are producing the product in a larger amount. So they are producing in a large fermenter tanks. Okay, so it may be in the tons. Okay, so amount is more greater than the amount that we are producing in the home. So that product, okay, will require large amount of time. Instead of 12 hours, it may require 48 hours. Okay, it may require 72 hours also. So, and how to recognize the product has been formed? See, incubation is generally at 35 to 37 degrees Celsius. Okay, this will be the temperature maintained. As in within this normal temperature only we can form the curd. Okay, now after incubation what happens? It has to attain a particular pH. So what we'll do here? Here we'll do the detection of pH. What is pH? That is negative logarithm of hydrogen ion concentration. That is called pH. So here that we are going to detect the pH, okay, of the particular product, and this pH after detection, that is slightly alkaline, okay, we can get the product is been formed. So it may be from the 6.8 to the 7.2, like that the pH should be there. Okay, and this pH range will confirm us that product is been produced. Now what we have to do? We have to withdraw the product. Next step is the withdrawal of product. Now here, what is the product? Whatever product we are supposed to produce. Suppose if we alter the temperature, we can produce other product also. But generally from this curd, yogurt can be produced and butter can also be produced. Okay, by churning. But cheese, for cheese we have to coagulate the milk protein. Okay, after coagulation we can get the different product like cheese, and different types of cheese are also there. For production of the different types of cheese in market, we know that pizza cheese is different. Okay, then some cheeses are there which are spongy. Okay, so different texture and different pattern of cheese will get in the market, and these cheese they are processed by the particular bacterium. Like Swiss cheese is there, which has been processed by the Propanobacterium shermani. This is the name of bacteria which is used for production of the Swiss cheese. Apart from this, Roquefort cheese is there. Okay, so these different cheese are there which are produced by the different bacteria, but they include the similar steps. Okay, then after withdrawal of the product, what will happen? The product is not purified, as it is consisting of the different bacteria, it consisting of the many bacteria. Okay, so what we are going? We are going under the purification process. Sorry, purification. Next step is the purification. So after purification, whatever desired product is there, they will be packed and stored into the refrigerator, or it will be sold into the market. Okay, so these are the general steps which are involved in the production of the dairy product. So for that, we have to pasteurize milk, we have to allow it to cool, then we have to prepare the starter culture and add into this medium so that we can get the desirable product. Okay, I hope you are getting this. So this is one of the example of the use of microorganism in the industries, that is in the food industry. Okay, such as dairy industry. Apart from that, in the food processing industry, we can also go for production of jam, jellies, pickles. Okay, then many other products are there which can also be processed by the bacteria. Okay, even though fruit juices, okay, generally the juices we are getting from the market, they are processed by certain enzymes. Okay, if we take an example, in the market we get different types of food. We have the fruit juices like Real, Tropicana, even though Epiphyse. Okay, that is the kind of they have the taste of the fruits. Okay, but there is no pulp inside it. Why? Because the pulp or the remnant of the fruit is been removed with the help of enzymes like lipases, proteases, okay, pectinases. As we know that like domestic or the herbivorous animals, we cannot digest the plant material directly, right? Why? Because in our body there is no secretion of the cellulose, sorry, cellulase and the pectinase. So what we have to do? We have to digest those material, okay, in the industries itself with the help of pectinases and the cellulase enzymes. So after that, the fruit juice which is available, okay, will be purified and packed into the bottles. So that is the enzymes which are used over there are also produced by the bacteria or some fungi. Okay, so these are the certain examples under food industry. I hope you are getting. If you have any kind of difficulty under this, you can ask me. For cheese, again, we can apply different temperature. Okay, just there is a slight change in the temperature for production of the different product. Okay, if we raise the temperature, we can get different product. Okay, there is alteration in temperature and the incubation period. Whatever time we are giving for incubation can also be varied. Okay, so this is about the food industry. Now next, that we will discuss about some other industries. Okay, so here, okay, in the beverage industry, let's discuss about the beverage industry example. Okay, so food and beverage generally we call together. Okay, so in the beverage industry, second, so in beverage industry, what we see, generally the beverages means what? The drinks. Okay, so it may include the alcoholic beverages also. Okay, so alcoholic beverages generally like beer, wine, vodka, gin. Okay, these are the different products of the alcoholic fermentation. Okay, so beverages can include the alcoholic beverages. Alcoholic beverages. Okay, so alcoholic beverages, okay, are of different types. It may be in the form of wine, it may be in the form of whiskey, or it may be in the form of wine, or it may be in the form of the distilled spirits, you can call distilled spirits. Okay, so wine can be used as a medicine in lower amount. Okay, it is used for treatment of various diseases again. And distilled spirits include the different alcoholic beverages or different alcoholic brands. Okay, can be now it is commercialized. Previously, it was used as a medicine only. Okay, so different distilled spirits include the beer, it includes the brandy, it includes the whiskey, rum, etc. Okay, these are the different brands. So these are under distilled spirit. Now, these alcoholic beverages, how they are produced? They are produced by the fermentation. Okay, produced by fermentation. So that is called alcoholic fermentation. And this alcoholic fermentation, it is being performed by a desirable microorganism. Okay, alcoholic fermentation is being performed by a fungus or a fungal strain called Saccharomyces cerevisiae. Okay, Saccharomyces cerevisiae, that is the yeast. Okay, generally we are calling it as a yeast. And other more strain of Saccharomyces cerevisiae is that Saccharomyces ellipsoides. Okay, Saccharomyces ellipsoides. So here, two different strains can be used for the production of wine or the distilled spirits. Now, in generally what we do, the wine or any distilled spirit, it is being produced from the malted cereals. Okay, malted cereals in the sense, see, cereal or grains are there, like barley grain. Okay, generally these products are produced from the barley. Now, barley, it is similar to the like wheat. Okay, so wheat, we know that gehu. Okay, similar to that, barley is there. And what we are doing, we are first of all the barley is being mashed, and after mashing, it will be mixed with the water. And in this process, we are adding a plant that is called hops. And this hops, what it does, it adds the flavor to this barley mixture. Okay, and after that, what we are doing, we are putting into the fermenter. Fermenter is generally a tank. Okay, it is a tank. So in this tank, there will be presence of the baffles, and these baffles, they are generally responsible for showing the rotation. So they are generally used for mixing the product. Okay, so it will be attached to the central part. These are generally the propellers, and they mix the, what they do, they mix the whatever product is there under this fermenter tank. There will be temperature sensing knob will be there over here. Okay, inoculating pipe will be there, from which we add the desirable product into, or the desirable fermenting substrate into it. Okay, here there will be ejecting tube will be there. After production of the desirable product, ejection will be there. Okay, this is a rough sketch. I will provide you the particular diagram for this. Okay, and there will be certain here insulating jacket is here. Okay, through which we can sterilize the medium before uses. Okay, so there are different methods. Okay, or different technology is being implicated here. So this is a typical structure of fermenter. What we are doing, we are preparing barley wort. After that, what we are doing, we are adding that mixture into the fermenters, and by adding this fungal strains, that is the yeast, that is Saccharomyces cerevisiae or the Saccharomyces ellipsoides, what do we do? We can maintain the temperature and incubate it for longer period of time, as we have discussed in the dairy industry, the steps. Okay, so first of all we sterilize it, afterwards we allow it to cool down, afterwards we have to add the starter culture prepared by this fungi. Okay, and then we have to add that starter culture into the medium so that it will ferment it, and after fermentation we will get the desirable product in the form of the alcoholic beverages, that may be in the form of wine, beer, okay, or whiskey, rum, etc. And these products, they are undergoing purification stages. So several purification steps are there through which we get the purified form of alcoholic beverage. Okay, so that is called distillation process. Through distillation we can produce the alcohol or the alcoholic beverages. So in this way, in beverage industry we are using the microorganisms. Okay, and as we know, the alcoholic beverages or any kind of other beverages, not only alcoholic beverages but also other beverages like soft drinks are there. Okay, or many fruit juices are also there, which are also been produced by the with the help of microorganisms. Okay, so here this is about the beverage industry and the useful nature of microorganisms in the beverage industry. Then we'll next talk about the pharmaceutical industry. Okay, next example that we are going to discuss is the pharmaceutical industry. I hope you are getting this. If you have any kind of difficulty, you can discuss. Okay, so next, in the pharmaceutical industry. So in pharmaceutical industry, for manufacturing of drugs in the sense the medicines. Okay, these are not the drugs which are taken in the film. Okay, so the drugs are the medicines which can be used for treating various diseases. Okay, so here in the pharmaceutical industry, again there will be implication of the what we say, the fermentation technology. Okay, so through fermentation technology we can produce a large amount of desirable product. Now here, what is the use of microorganism over here? See, as fermentation is concerned, microorganisms are also applied over here. Okay, let's take an example. So here we will discuss about a product that has been produced by a new emerging technology called biotechnology. Okay, that is the recombinant insulin. Okay, example here is the recombinant insulin, or we can also call it as a genetically engineered insulin. Genetically engineered insulin, or it is also called as humulin. Okay, so the production of the insulin was been proposed by an American company, that is Eli Lilly. Okay, it was initiated by an American company, Eli Lilly, and it was a pharmaceutical product. Now, what is the need of production of this insulin outside the body? As we know, the main purpose of the insulin is to maintain the blood sugar level, right? So if the blood sugar level rises, then that will be regulated with the help of, or it will be lowered with the help of insulin. But in our body, due to certain disorders or due to certain malfunction, we are unable to produce the sufficient amount of insulin. So we suffer from a disease called diabetes. Okay. So to overcome these diseases, what scientists have done, they have isolated the genes. See, hormone or the insulin is a peptide hormone. Peptide in the sense it is a proteinaceous hormone. So proteins, how they are synthesized? They are synthesized from the DNA, that is the genes. Okay. So here there are certain specific genes which are required for production of this insulin. Okay. So the genes which are required for production of insulin, they are isolated. So here, what the steps will be there? First of all, see, we have to go through the recombinant DNA technology, and recombinant DNA technology is nothing but a selection, isolation and insertion of the particular gene into an appropriate host with the help of suitable vector. Okay, whatever I am telling, just focus on that. Selection, isolation and insertion of a desirable gene into a appropriate host with the help of suitable vector is called recombinant DNA technology, or it is also called genetic engineering. Now here there are certain steps which should be followed under this process. So first of all, what scientists has done, they have identified the gene. Okay, suppose this is a human DNA. Okay. In the human DNA, particular this region, it is responsible for production of the insulin hormone. Means there is a segment of DNA, that is gene, and that gene is producing the insulin hormone. Now what scientists have done, they have isolated this gene. So first step is the selection, particularly they are selecting this part. So first step is selection of gene. Then second, isolation of gene. Now isolation of which gene? Gene of interest. Okay, so here we know that this gene, okay, is supposed to produce the insulin hormone. Now what we are doing, we are cutting this gene with the help of the enzymes. Okay, so generally endonuclease enzymes, okay, used to cut DNA. Okay, so endonuclease enzymes are used to cut the DNA segment. Now after cutting, we are what we are doing, we are isolating it. That means we are separating it. Okay, then third step is insertion into the host, but before that we have to insert that gene or fragment of gene into vector. Okay, so what is the vector here? See, in the bacteria generally there is a extra chromosomal set of DNA. We have studied about it, that is called plasmid. Okay, so here what we are doing, we are, suppose this is a plasmid. Okay, this is a plasmid. It is double circular DNA, and if you are unable to observe here, you can see here, this is a plasmid. Okay, so this plasmid consists of right border, left border. Okay, and there is certain place which is called MCS, that is multiple cloning site, and this is the site for addition of that gene. So this gene, whatever we have selected, will be added over here. Okay, so it will be added over here. So when the plasmid, and plasmid has a property called autonomous replication, it self-replicates. So when it replicates, it will also replicate this desirable gene. Now, where exactly this plasmid is going to replicate? It requires a cell, right? So it will replicate within the bacterial cell. Okay, now here we are inserting the gene. Okay, insertion into vector. Insertion of what? Desirable gene into the vector. Then afterwards, insertion of vector. Okay, so generally plasmids are used as the vector. Plasmid, bacteriophage are there, phagmid are there, then cosmids are there, different lambda-phage is there, different types of vectors can be used. Most common type is the plasmid. Okay, so insertion of vector into bacterial cell or bacteria. Okay, after insertion of that plasmid into bacteria, it will multiply. Okay, and after multiplication, it will produce the desirable product. Okay, so fifth step will be product formation. So as the product is formed, this product, what it will be done? This product will be purified as it is produced within the bacteria. Okay, this product will be purified and will be what we say, inoculated into the vials and sold into the market in the form of medicine, or it will be packed and sealed into the pharmaceutical industry. So, and why? Generally, why we are using bacteria for production of this what we say, insulin? Because bacteria, they can replicate within 20 minutes. Okay, so a single bacteria such as Escherichia coli, okay, E. coli, they replicate within 20 minutes. So suppose we have thousands of Escherichia coli, and each E. coli or the bacteria can produce the insulin. Okay, after addition of that gene into that bacterial genome. Okay, so suppose 1000 bacteria are there, 1000 times we are producing the insulin hormone. Okay, so in this way, in much higher amount the insulin is being produced, because bacteria, they require what? They require artificial nutrient medium. Again, so whatever product we are getting here, okay, that will be the recombinant formation of product formation, that is the recombinant insulin. Okay, and this recombinant insulin is been again processed under fermentation method. So now bacteria, they consist of the desirable gene, and this desirable gene is produced in higher amount by the fermentation, and in this way we get the larger amount of insulin. In similar way, many different medicines can also be produced. Not only the insulin. If we take an example of antibiotics. What are antibiotics? These are the chemical substances which inhibit the growth of another pathogenic microorganism. Okay, so the first antibiotic was discovered by Alexander Fleming, okay, in the year 1929. Okay, so he has produced the first antibiotic as a penicillin from the Penicillium notatum and the Penicillium chrysogenum. So these are the two different steps by which penicillin was produced. Now, what is the use of that penicillin? Okay, it was acting as an antibiotic. So it was being used, okay, by the soldiers for first of all. Okay, then it was again being modified. Many different antibiotics were synthesized. Now, how bacteria synthesizes the antibiotics? Suppose these are the two bacteria. Suppose this is one bacteria. Okay, we'll say bacteria A, and this is the another bacteria, bacteria B. Now, see, all these bacteria, they require a survival. For survival, what they do? They releases a chemical around them. Okay, and this chemical, it is providing a area. Under this area, another bacteria cannot enter. Right? So that is the zone of inhibition, and that zone of inhibition is nothing but a production of antibiotic. Anti means against and biotic means life. So against the life, chemicals are secreted. So it may be for similar type of bacteria again. Okay. So what we are applying the antibiotics, when we are applying antibiotics to our body, whenever we are getting any kind of infection. Okay. Suppose it may be the infection of tooth, it may be the infection or the wound we are getting. At that time also we are taking the antibiotics. Right? Why? Because that wounded part of the body may get affected via certain pathogens, and pathogen means the disease causing organisms. Okay, so this disease causing organism may have a kind of lethal type of effect on our body. To prevent it, we are applying the antibiotics. Okay, apart from that, we can also produce many different types of products. Okay, through the pharmaceutical industries. I hope you are getting this, because as I am taking very important examples. Okay, what we have to do, I'll provide the notes after the lecture. Okay, so you have to write down those notes and study those notes. If you have any kind of difficulty, you can ask me. Now, apart from this, another example that we are going to discuss is the biological control. Okay, or as the use of microorganism as a biological control agent. Okay, so let's see. So generally, biological control means what? We are implementing the microorganism or the use of microorganism in the field of agriculture. Okay, so in the field of, as we know, not only India but most of the countries, they are based on agriculture. Okay, so agriculture is the main source for the food material that we get. Okay, and we know that from the agricultural land we get the various nutritious food material. So to maintain their nutritive value, okay, as well as to maintain the proper food quality or the fruit quality, what we have to do is we have to produce the efficient type of food product or the fruits. Okay, so how to produce that efficient products? By use of the biological inoculant. So here we will take the example next, that is the biological control agents. Okay, or you can also write biocontrol agent simply. Okay, so what is the use of the biological control agent is, okay, it can act as a biofertilizers, okay, or it can be in the form of biopesticides, or bioinsecticides. So biofertilizers are nothing but a microbial inoculants, are the microbial inoculants. Okay, now what is meant by microbial inoculant? First of all, we will understand this term microbial inoculant, then we will understand what is biofertilizer. Okay, so fertilizer, we know that increases the fertility of the soil, but here biofertilizer means what? The fertilizer which are produced by the living organisms, okay, are called biofertilizer. Now microbial inoculant, why we are calling microbial inoculant? Because the fertilizers which are produced by the living organisms generally involve the microorganisms. Okay, so see, in the soil there is a presence of micronutrient and macronutrient which are required by the plants for the growth as well as development. So here for growth and development they require nitrogen, they require carbon, they require the phosphorus, okay, they require magnesium, manganese, iron, copper, okay, and boron, like zinc is also essential for them. So many different mineral or the nutrient they are required in larger amount. Even calcium is also required. So these all nutrients they are present within the soil, but sometimes due to some reason plants are unable to accumulate all these nutrients, okay, directly. So what we do is we will implement the microorganism which keep a kind of association with the roots of the plant and fixes the atmospheric nitrogen, fixes the various nutrients into the root of the plant. Okay, so such microbes, okay, can be used as the biofertilizer, and such a microbes are also called as the microbial inoculant. Inoculant means we are inoculating that bacteria into that soil, okay, or onto the plant or the crop material. Now the process for production of the biofertilizer, again it is the fermentation method. Okay, again it is the fermentation method, as we can select the different types of biofertilizers or the different types of microbial inoculant. Okay, let's take an example of the certain microbial inoculant such as rhizobium. Rhizobium. Now what is rhizobium? It is a soil gram-negative bacterium, okay, which is present generally in the root nodules. So suppose this is the root part, okay, and to the root part generally there will be presence of the root hairs, and this root hairs may have the large bulb-like portion at the tip which can be called as the root nodule, okay, which can be called as the root nodule, and generally found in the leguminous plants, okay, or the leguminous crops. So this rhizobium, it is been situated in this root nodule. Now what we have to do is we have to extract the root nodule from the crops. We have to process under laboratory by providing the artificial medium. Again we have to go for fermentation. Okay, so from a single bacterium we can produce larger amount of bacteria. And now the generally generally the medium used for production of rhizobium is the yama. What is yama? Yeast extract mannitol agar. This is an artificial nutrient medium under which the rhizobium can be cultured, and after culturing rhizobium we can produce under the laboratory conditions in a larger amount under the fermentation tanks. Okay, apart from rhizobium, some few more examples are there like Azotobacter, okay, which is also responsible for fixing the atmospheric nitrogen as well as helpful in the fixation of the various important minerals. Apart from that, we have the Pseudomonas, okay. Apart from that, we have the azospirillum. Okay, apart from that we have the nostoc, anabena. These anabena, nostoc are the cyanobacteria. Okay, this can be also used or implemented for production of the desirable product by the plants. Okay, so what they do, they fixes the nutrients within the root of the plant, so that plant get fertility factor and they can produce the good products. Okay, then micronutrient and macronutrient, they play a great role in the development of the plant. Okay, so yellowing of leaf is also due to the process called chlorosis or the necrosis, that is loss of chlorophyll. Okay, or the necrosis means it is a accidental cell death. So loss of chlorophyll is due to the lack of nitrogen. So nitrogen should be provided in appropriate amount to the plants. Then biopesticides can also be produced in the industries. Okay, and these biopesticides can be produced by the help of again recombinant DNA technology. Okay, so these are some examples of the biological control agent. Okay, so here we can maintain the fertility of the plant, and here we can maintain the insect and pest resistance. Okay, so this is about certain examples. In again, apart from that, enzyme is there. Another example that we are going to discuss is the enzyme production. Okay, so enzyme means what? Enzymes are the biological catalyst. Okay, so fifth, enzyme production. Okay, so enzymes generally they are acting as a biocatalyst. So biocatalyst means those which can accelerate the biological reaction. Accelerate the biological reaction. So here we can see the biological reaction, they are getting speed up, and as the biological reaction they are getting speed up, we'll get the product very faster. Okay, so enzymes they accelerate the biological production. Okay, so here the different enzymes are there. Different classes of enzymes can be studied, like the enzyme which can perform, like we have 6 different classes here. One, that is the oxidoreductase. Okay, which brings about the redox reaction. Or the oxidation and the reduction reaction simultaneously. Okay, then second is the transferases. What is the function of transferases? They are helpful in the transfer of a functional group. Then third one is the hydrolysis. This is bringing about the hydrolysis. Then fourth one is the isomerases. Fourth one is the ligases. Okay, that means they are responsible for joining. Then fifth one is the isomerases, and sixth one is the lysis. Okay, so lysis they bring about the breakdown. Isomerases they are responsible for formation of the isomers. So generally these are the different 6 class of enzyme, can be remembered as OTLIL. OTLIL. Okay, so here these enzymes they are basically used for various different biological processes, and these biological processes they may helpful in production of desirable product in the fermentation industry, in the alcoholic beverages, in the food processing industries, okay, even in the pharmaceutical industry, different types of processes are. In the textile industry also, okay, different types of enzymes can be implemented. Now, for production of this enzyme, microbes can be used. Different microorganisms are there, okay, which are responsible for production of the enzymes. Okay, so these are the certain examples, okay, of the useful microorganisms in the industries. Okay, in the next part we will discuss about the environment management and the disaster management. Okay, thank you.